Felistus Mulwa. I'm a graduate fellow, PhD. I'm registered at Sokoine University of Agriculture, Morogoro in Tanzania, PhD in mathematics, statistics option. I'm sorry, my topic changed a bit when I came to ILRI. I'm here for my sandwich program between ILRI Animal and Human Health Program and the ISIPE Partnership in Applied Sciences, Engineering and Technology Scholarship, PASET. So I'm going to present on a proposal on modeling and assessing the impact of Rift Valley fever disease outbreaks on food security in Isiolo County, Kenya. That's my presentation outline. Sorry for that. I started my sandwich program this year, January, so I'm presenting my proposal. So Rift Valley fever virus, or RVF, is caused by Rift Valley fever virus in farmed animals. The outbreaks are usually very common in the pastoral areas, which are usually inhabited by pastoralists, and its outbreaks usually result into great extensive economic losses and also public health burdens. The disease also has the capacity of attacking human beings. The virus was first identified in Kenya, that is 1981, in Naivasha, and the first animals to be attacked were the sheep. And since 2015, the disease has been listed as one of the priority emerging disease by the World Health Organization. Since then, again, outbreaks have been reported outside Africa, that's including Yemen and Saudi Arabia. In the last 4 years, periodic outbreaks of RVF have been reported in the northeastern Kenya. So on the statement of the problem is that the frequency of these outbreaks have increased in the recent past. The first major outbreak was the 1997-1998 outbreak. So multiple outbreaks have been reported, and there is no information on how to link the RVF with human nutrition impacts. So my research aims at developing a statistical modeling approach to quantify these impacts of the RVF and human nutrition. When RVF occurs, it has the capacity of disrupting the food systems, and hence it threatens the food security. Children, the aged, and women of reproductive age are usually threatened due to their nutritional requirements. My study targets the children under 5 years. So far, there's no evidence on the effect of these outbreaks on food security in children, and also the response to these future outbreaks, that is in Isiolo County. Also, I have 4 objectives. So the general objective is to model RVF outbreaks and assess their impact on food security and human health in Isiolo County. I have 4 specific objectives. And the first one is to develop a statistical model for predicting RVF outbreaks. The second one is to determine the effect of RVF outbreaks on food security in Isiolo. The third one is to investigate the effect of the RVF virus on dietary diversity. And the last one is to investigate the effect of RVF on the nutritional status and health in Isiolo County. So for my objectives, RVF is usually favored by climatic conditions, especially excess rainfall. So most of the studies usually link they usually link RVF with climatic conditions, but very few has been done to link RVF and food security and then human nutrition. So I have 3 research questions which are derived from the specific objectives. And the first one is, how can we accurately predict RVF outbreaks using statistical approaches? And what are the effects of RVF outbreaks on food security? And the final one is, how does the knowledge of these RVF outbreaks influence the response and hence food security and human nutrition? The study site and population. So the study site is Isiolo, which is northern Kenya. So I have 3 study sites, that is Garabatula, Marti, and Sericho. So these areas were purposefully chosen following a previous RVF outbreak, that is December of 2020. So on the first objective, which we'll touch on time series modeling, we'll use historical data sets, and this data will be obtained from the ongoing co-infection project, whereby I will use the longitudinal data. The project is based in Kenya. I'll also get the data set on RVF outbreaks here at ILRI. I will also get data on food prices from the World Food Program, because whenever we have an RVF outbreak, the prices of the food prices usually go high. I'll also get additional data on RVF outbreaks and vaccination from the Isiolo County Department of Agriculture, Livestock and Fisheries. Mainly the dataset will consist of the RVF cases, the monthly and yearly outbreaks. So for the first objective to measure the RVF outbreak over time, I will develop an interactive dashboard using IR-Shiny Hub, whereby I will capture the trend of the disease and the climatic patterns. These climatic patterns will majorly be composed of the rainfall, whereby which we all know that excess rainfall favors RVF outbreaks, and other characteristics, including latitude, altitude, topography, and also the slope of the area. So for the first model I will be working on is the autoregressive integrated moving average model, which is a statistical model which is usually used for prediction. This model will be applied to the secondary data. This is how the model looks like, whereby it has several parameters. These parameters will be obtained from the data sets. So once the model is fitted, this is the flowchart for the model selection, or how the steps of how I'm going to fit the model. So starting from removing the noise or irrelevant information from the model identification, the estimation of parameters. The parameters will be estimated from the data set, testing the model, validation, and finally forecasting. So on the second objective, still on modeling, I will develop an intervention time series model, which is a statistical model, to model the RVF outbreak whenever there is an intervention. So this picture here shows how the model will work, whereby we'll have the normal flow, then the level change whenever we have an outbreak, then the observed. So all these slopes, I'll get the level of change, and also the change in trend, which I'll incorporate the variables to fit the model. So that's how statistically, that's how an intervention time series model looks like. So on the third and fourth objective, whereby I will be linking the RVF outbreaks to food security. So food security has 4 components. That's availability, accessibility, stability, and utilization. So parts of these components are usually affected whenever we have an outbreak. So for those objectives, I'm going to get primary data on the field on top of the historical data set, so that I can clearly investigate how RVF affects any of these components, especially availability and utilization, since I'm working on children under 5. So for the statistical analysis, I'll fit several models, including the ordinal logistic regression, the multiple, mixed methods analysis, and also analysis from the retrospective data sets, sensitivity analysis, so as to link the RVF to food security and human nutrition. So that is what my third and fourth objectives will look like. So the data will be a cross-sectional study to investigate the components of the food security, whereby an household questionnaire will be administered, and the sample size will be calculated, 286. So the ethical documents have already been obtained from the IREC and the National Council for Science and Technology, NACOSTI, here in Kenya. So the main components which I'll be investigating are the household food security in access scale, which usually it's a scale which shows the degree of insecurity for the household in the previous 4 weeks. So it will be classified into to 4 categories. That's the food secure, that's the normal, slightly food insecure, moderate, and severe. Then the other component will be the household dietary diversity, which is a score that ranges from 0 to 12. But since I'll be studying children, the food groups usually reduce to 7 or 8. So I'll investigate the dietary diversity score and link that to the impact, how it is affected by RVF through the statistical modeling. So the relevance of this study will help in contingency planning, sensitization, early warning signs, and better handling of future outbreaks of RVF and food security. So those are the selected references for my work. So I acknowledge ILRI, the Animal and Human Health Program, and the RVF Nutrition team, the sponsors of my scholarship, that is the ISIPE, the host university, the Sokoine University of Agriculture, whereby I'm hosted under the Success Foundation for One Health. My supervisors, the ILRI CapDev team, I acknowledge all those. Thank you.
Thank you very much, Damaris, for that clear presentation. And just a general comment to the presenters, I think it's good when you're introducing yourself, if you can put your camera on so that we see who is presenting, and if you feel it's degrading your your internet, then you can switch it off as you move to your presentation. So before we move to the next presenter, I want to give this opportunity to your supervisors, if we have any online, because they might be maybe wanting to leave after your presentation to make comments, if any. Not sure whether you have any.
Yes, Nicholas. Thanks for
yes, go ahead, Bernard. Yes.
Yes, I think Damaris has really done a very good job in describing the study, so I don't have much more to add, but also to say, given that it's a new area, really not many studies have been done to show how livestock diseases affects human nutrition. So lots of those models which she's talking about will have to be refined, and we select a few that will be used for quantification purposes. So yeah, so this is a start, and I'm happy that, we are at this stage.
Thank you so much, Bernard. And yeah, I think tied to that is a comment I wanted to make in terms of simplifying the slides so that, alongside those equations, we also have things, the general audience can understand. So thank you so much, Bernard. Do we have any other supervisor? And moving on, I would want to request that we allow Isaac Kasoro to go first, because the supervisor has requested that, because she has another commitment after this. So we have Isaac going next, and he is going to present on comparison of pig semen extenders in Uganda. And his ILRI supervisor is Karen Marshall. So Isaac, please share your screen. I let you know once I see it from our end. Put it on slide view. Isaac? Isaac, can you hear me? Isaac,
Nicholas,
Isaac, we are struggling to hear you. I guess probably it's your network.
Hello, Nicholas.
Yes, good. Now
yes,
we can
Nicholas,
hear you.
I can hear you.
Yes. Now we can hear you and we can see your screen. So yeah, go ahead. And if you feel
Hello,
your
Nicholas,
video is
can you
going
hear
to
me?
affect the internet, just switch it off. Switch the video off. And go ahead. Isaac, we lost your screen. Try to share again. There we have it. Put it on slide view and take over.
Okay, good morning once again. I confirm if you can hear me and you can see my screen.
Yes, we can hear you loud and clear and you can see your screen. Go ahead.
Okay, sorry for that. My name is Kasoro. I'm a graduate student at Makerere University, pursuing a master's in animal science. My topic is the evaluation of porcine semen extenders affecting smallholder farmers' access to quality semen in Uganda. My supervisors are Dr. Karen Marshall from ILRI and Professor Donald Kugonza from Makerere University. Today, I'll give a brief introduction about my work, the problem statement, how I intend to do my work, the conceptual framework, the objectives, the significancy, and the materials and methods that I intend to use. Currently, I'm just getting ready to start my work. I'm just finalizing with the data collection tools and the proposal. I'm still discussing with my supervisors. Now, the pig industry is quite important in the Ugandan sector and contributes to, actually supports 1.3 million of the households in the country. But it's important to note that 90% of the pig farmers in Uganda are smallholder farmers rearing at least 1 to 5 sows or pigs. And but since they have very few animals, even their production systems are also quite enabling them to rear these few animals. Majority of the systems, the production systems they employ are the tethering system, the small semi-intensive and intensive systems. So in these systems, they can only support a variety of breeds, majorly crossbreeds. For example, we can have the crossbreeds, the exotics, and of course the local. And you can see on my right here, I've tried to show the different that you're likely to encounter when you're dealing with the Ugandan pig industry. Up the top right, the different production systems in Uganda. This is under a tethering system. Then you can have the local pigs, this down, top left corner. Then of course, this could be crosses that can also be used to, you're likely to encounter when you're dealing in the Ugandan sector or the piggery sector in Uganda. So since we have very few smallholder farmers and they're having few animals that they're keeping, so even the production and the productivity of the pig industry is still low and is still manned by a lot of challenges. So in this regard, pig artificial insemination technology is one of those technologies that was highly attributed or shown capacity to improve the production sector. But however, we're also facing challenges with this technology And this could be aligned to the low use and adoption of the technology in Uganda. This could mainly be from the low cell conception rates, poor heat detection by the farmers by the time they call the AI technician, the heat yet, and the AI technician is forced to inseminate maybe a pig that is not understanding it, or maybe probably not fully on heat. So that culminates into increased number of repeats and ultimately low conception rates. But there's also knowledge gaps, and this knowledge gap could be between the farmers not clearly knowing the advantages of pig artificial insemination compared to natural mating, and also the transfer of knowledge from people who know the knowledge to the farmers is also lacking. So there's a huge gap within that needs to be addressed. But also there are also issues of semen handling, semen processing, and semen transportation to these different units that aids into these low conception rates and the rest. So in bid to address this, there was a variety of extenders on the market, but as you can see on the map here, we have a few semen producing units that are located within the central region, and these semen producing units have to move semen, send semen to the farthest regions within Uganda. So it is known that as we're moving semen, we're going to be losing its viability, liquid semen. It's going to lose its viability and quality as we move it over these longer distances of transport. So we need to have an understanding of the extenders and how best they can be able to keep this semen, and then we are able to achieve successful conception and viability of semen even across the long distances, or the longer distances and the time they have to be moved. So this is the baseline for my research. We need to, I'm looking at evaluating the different extenders on the market and how to be able to suggest the best candidates that can be employed by these few semen producing companies that are already centered within the central region to be able to supply to as majority of farmers within the Ugandan sector as possible. To carry out this work, I intend to follow the conceptual framework as shown below. I will have a laboratory analysis of these different semen extenders in the market, and this will be done at Makerere University Research Institute in Kabanyoro and the Animal Science Lab. Being able to list up to a tune of 30 extenders that can be broken down to short-term, mid-term, and long-term extenders. But however, I cannot do this evaluation of extenders without value chain majorly focusing on these semen producing units. These will give information of the key semen production aspects, marketing, and all those attributes that now lead into improving the semen production. I also intend to have a few semen samples from the semen producing companies and use them in the comparison and selection of the best candidates to use. And ultimately, I hope to extend the boar shelf life in the country. But to achieve this, I have the objective, which is strengthening the adoption and use of pig artificial insemination technology in the country through increased farm access to good quality extended boar semen, by improving the extension procedure and handling of the semen. To do this, I'll need to evaluate the pig genetics value chain with major emphasis on the production aspects, boar genetic diversity, and marketing attributes of these key or different semen producing companies, as well as evaluating the different commercial extenders we have on the market and how to improve them to determine the best use for our country. The importance of this work is it will also contribute to the different efforts that have been carried out in strengthening pigs, like reduced disease outbreak, increasing biodiversity, giving the farmer an opportunity to select the genetics of their choice, among others. So we will be contributing to these efforts and strengthening the development of the pig industry in Uganda. Also, this technology can also provide a source of employment for not only people who are offering the service as AI technicians, but also the emerging sectors where we have people who are going to be supplying the AI consumables and, of course, among others. So ultimately, when we are able to achieve this increased semen viability, we have to increase farm production and farmer access, and ultimately increasing the pig industry and livelihoods of the people. For achieving my first objective, which which is mapping of the pig genetics value chain. This I will conduct this majorly within the central region of Uganda and within the 5 districts in green on this map. And why do I select the central region? Accounts for majority. The central region accounts for majority of the pig production in the country. And also it accounts for close to 19% of the total number of pigs we're having, but also majority of the semen producing companies we have in the country are all centered within the central region. Conduct a census for all the semen producing companies because there are quite few. I cannot now limit on the sample size, and then the size that I will be able to work with will be limited to the access and willingness for these to participate in the study. We'll have structured questionnaires that will get my key informant interviews where we'll have discussions with these semen producing companies to be able to get information on semen production aspects, majorly the boars they have, the boar diversity, how they source the boars, the different extenders they use, the collection intervals per boar, and then of course the selling and then the disposal, the marketing channels, and of course how they get information about semen handling, how they get information about improving semen practices and all that. Then I will do analysis for both the quantitative, which is explaining what they do, and the qualitative in terms of the doses they produce and the rest. The second objective, I will do an evaluation of the different commercial extenders, and this will be done in Kabanyoro. We shall collect, the semen will be collected from the boars using a gloved hand technique, and this will be done by a trained technician, and we shall collect from at least 8 boars. That is accounting for at least 2 from each breed. I have chosen the Duroc, the Landrace, the large white, and the Canberra lines because those are easy to, they are the most common breeds that are being used, and I will dilute using the 30 different commercial extenders that I've been able to list, but however, this number can vary depending on the evaluation of the pig genetic value chain that will happen. And these extenders can be categorized short-term, mid-term, and long-term. And as I stated, I will get some liquid semen samples from the different semen units to use for analysis. Then each ejaculate that will be collected will be subdivided into 30 different equal parts, and then any will be randomly diluted using the ratio of 1 to 4 in any of the 30 extenders, and then stored at 17 degrees Celsius because this is the recommended temperature. Observation will start immediately after dilution, that is at 0 hours, then 1 hour after dilution. The instance of 1 hour is to see the response in terms of the buffering ability, the motility, and of course the DNA integrity at that level, and then 12 hours post dilution. Then the analysis will continue at day 1 up to day 15, and each day I will analyze in the morning and the evening, that is in a 12-hour interval. Basically, I'll be focusing on differences in mortality, and these differences will focus on the average path velocity, the average path velocity, the linearity, and the progressive motility, as well as the straightedness of the semen. I will also conduct a hyposomosmotic test for membrane integrity, and I will do analysis using the R software. I would like to acknowledge Chaplin for funding my work, Dr. Karen and Professor Kogonza from ILRI and Makerere respectively for the support rendered in developing the work so far, and the ILRI CapDiv team for allowing me to present, and all of you for attending. Thank you very much.
Thank you so much, Isaac, for that very clear presentation. I'll pass it to Karen if she has something to comment.
Yeah, thank you so much, Isaac. That was a great presentation. And yeah, just to comment to all that this is part of a larger piece of work that we're doing under Sapling, really to strengthen the pig AI system in Uganda. And this constraint of limited semen life has been raised, for many years by various stakeholders. So we're very glad to have Isaac actually working on this issue in a sort of direct industry relevance. And at the end of it all, we will give all of this feedback during workshops to our industry partners, and hoping that we'll have something that they'll be able to use. So yeah, well done, Isaac. Thank you.
Thank you so much, Karen. Now, please, the participants continue putting your comments and questions on the on the chat box. So we'll move to Enock Kiprono from the Animal and Human Health Program. Is going to present on seroepidemiology and molecular characterization of Coxiella Burnetii in livestock in Isiolo County. His supervisors are Bernard Bet at ILRI and Hussein Abkalo. So over to you, Enock. Please share your screen and take over. Put on slideshow. Good. Now you can go ahead. You can see your screen. Of course, I can't hear you.
Hello.
Yes, good. Go ahead.
Okay. Hi, everyone. My name is Enock Kiprono. I'm a student at Jomo Kenyatta University of Agriculture and Technology, taking masters in molecular biology and bioinformatics. So I'm MSc graduate fellow here at ILRI. So I'll be presenting on seroepidemiology and molecular characterization of Coxiella Burnetii in livestock in Isiolo County, Northern Kenya. My supervisors are Dr. Bernard Bet from ILRI, Dr. Hussein Abkalo from ILRI, and Dr. Joel Barkul from JKUAT. So I welcome you to my presentation. So livestock farming is one of the key pillars in an economy. In developing countries like Kenya, many livelihoods are dependent on livestock farming to boost their living. So globally, livestock markets employ more than 1.3 billion people and boost the livelihoods of over 600 million of poor farmers in developing nations. However, the growth and stability of livestock farming is affected by many factors. Key among them are the climatic changes and zoonotic diseases. These factors consequently affect livestock productivity and the livelihoods that are dependent on livestock farming. So globally, there are over 1,400 zoonotic diseases that are acquired by man directly from animals or vice versa, contributing to 4/5 of human diseases. Q fever, caused by Coxiella Burnetii, is a worldwide neglected zoonotic infection that affects both animals and humans. So the host of these bacteria include domestic and wild animals, birds, and arthropods. Sheep and goats are the main reservoirs and the source of infections for humans. So ruminants may develop chronic diseases and symptoms may include abortion, delivery of premature offspring, steel parts, and weak offspring. The shedding routes in infected animals include part products, semen, urine, and vaginal discharge. So 75% of the infectious diseases are zoonotic in nature, which are attributed to factors such as religious and cultural lifestyle. Zoonotic infections have a significant impact on both public and animal health. Impact on the public health are categorized as direct or indirect implications. Direct implications are the consequences in terms of increased morbidity and mortality. Indirect are the effects of the consequences on the general public. So the impact on animal health manifests itself through increased abortion cases, steel parts, delivery of weak offsprings, in livestock. So despite the fact that Q fever has not been prioritized as a serious cause of animal and human diseases, studies done in Kenya have reported the prevalence of Q fever in animals and human population. Q fever has been listed as one of the zoonotic diseases that cause socioeconomic burden in marginalized pastoral communities. For justification, due to close proximity with livestock, pastoral communities stands at a high chances of acquiring infection, which manifests itself as a febrile illnesses in humans. Febrile illness in humans is normally misdiagnosed. It is treated as a presumptive malaria, which results in missed opportunity to accord, identify, and treat other cause of the fever. Therefore, Q fever can be misjudged for other febrile illnesses in humans due to lack of awareness among health personnel and contribute to decreased ability to diagnose Q fever. So this study is important as it seeks to improve on laboratory detection of Q fever, which is one of the biggest challenges that has been identified in the analysis conducted by the stakeholders on the prevention and control of zoonosis using One Health approach. So in view of the Sustainable Development Goals on good health and well-being, this study will help to provide more data about the aforementioned pathogen to reduce the risk, thereby contributing to risk management both nationally and globally. So I intend to answer the following research questions. The first one is, what is the seroprevalence of Coxiella Burnetii in aborted livestock in Isiolo County, Northern Kenya? And the second one is to answer the question that, what is the genetic diversity of Coxiella Burnetii? So here are my research objectives. My general research objective is to characterize and determine the seroprevalence of Coxiella Burnetii that cause abortion in livestock in Isiolo County, Northern Kenya. Specific objectives to determine the seroprevalence of Coxiella in aborted livestock in Isiolo County. The second objective is to assess genetic diversity of Coxiella sequenced from livestock in Isiolo County. So since I'm dealing with infectious pathogen, then I will put in place the biosafety measures. And one of them is to work in a biosafety. I will be working in a biosafety level 2 laboratory, as Coxiella poses a risk both to the handler and the environment. So here are the biosafety measures that I'll put in place. I will use the appropriate personal protective equipment such as gloves and lab coats. Also, appropriate decontamination measures and practices such as appropriate waste disposal. Also, maintenance of sterile working conditions such as working in a biosafety hood. So my study site is Kina Ward, that is in Karapatula Sub-County, Isiolo County. Study design is longitudinal. Sampling method is opportunistic sampling. So the sample size for my study is for my serological analysis, that is my first objective. I will work with 237 samples. So to achieve the sample size, I used the formula that I have projected here to work out the sample size. So when I was working out the sample size, I used a prevalence of 3% in cattle, an average prevalence of goats and sheep of 14%. So I used a study that was done in Tana River and Garissa counties. And since the study was done under pastoral setting, I chose to use their prevalence in my study, since I'm also doing a study in a pastoral setting. So here is the workflow of my methodology. The first one is ELISA, then molecular analysis, then finally phylogenetic analysis. So for my first objective, I will do ELISA. I will run IgG ELISA to detect the presence of antibodies for Q fever. So that is how I will achieve my first objective. So for my second objective, that is the molecular analysis. and also phylogenetic analysis, I will use the DNA extraction kits to extract the DNA from the collected placental tissues. Then for PCR analysis, qPCR, I will use qPCR to detect the presence of Coxiella using specific primers. Thereafter, the samples that are positive for Coxiella in the qPCR, I will use it to run a nested PCR using primers for multispacer typing of Coxiella. So after that, I will do gel electrophoresis to visualize the amplification and as well as to estimate the size of my bands. So continuation of my molecular analysis. So the PCR products will be first purified using the purification kits before doing the sequencing. So here sequencing is a prerequisite step for phylogenetic analysis. So for sequencing, I will do sequencing using the primers for multispacer typing of Coxiella. So once I'm done with the sequencing, the nucleotide sequence obtained from sequencing will be first edited using bioedit software. Then the edited sequence will be compared to reference sequences that will be retrieved from the gene bank. The edited sequence and the reference sequence will be subjected to multiple sequence alignment using MASO software that is embedded in bioedit. So finally, I will do phylogenetic analysis and I will generate a phylogenetic tree using maximum likelihood approach on Mega software. Then in order to ascertain the reliability of my phylogenetic tree, I will do a bootstrapping analysis of the replicates. Then also, finally, in order for me to visualize the phylogenetic tree, I will use fig tree to visualize. So for my data analysis, data will be first cleaned and managed in MS Excel before proceeding to R software for analysis. So R program, I will use it to determine the positivity rates from serological analysis. Then for ethical approval, so my project is nested on a bigger co-infection project. So I will use the ethical approval for the bigger project, that is the co-infection project. So here is my work plan. I intend to finish my work this year, November this year. So here is my budget. Acknowledgement. So I would like to acknowledge the following partners in this project: ILRI, Government of Kenya, Zoonotic Disease Unit, JKUAT, Kenya Wildlife Services, Washington State University, University of Nairobi, and finally Los Alamos National Library. Thank you.
Thank you very much, Enock, for that clear presentation. Again, today we had really wonderful presentations. Now I'll open the floor for any questions. Just raise your hand and ask the question to any of the presenters. Then I'll finally read the comments or the questions on the chat. Over to you, the audience. Anyone with any question to anyone? As you get ready, I'll read some of the questions on the chat, and one goes to Damaris. Philly says, great presentation. Do you have an hypothesis for your second objective? The objective is the effect of RVF outbreak on food security. I saw you try to reply, but you can unmute and just respond. Damaris?
Yeah, thank you, Nicholas. Yeah, I've already replied in the chat. So on that specific objective number 2, I'll be investigating whether RVF really affects the 4 components of food security, that is availability, utilization, stability, and stability. That's children under 5. I think that's their policies.
Okay. And to you, Damaris, I have some comments, and one of it is what I meant earlier, that try to simplify, have, like, the graphs were nice so that at least it explains those formulas you tried to use there. And then, of course, when you are presenting or on the slides, avoid those abbreviations, which we don't know what they mean. You mentioned, fine, the one on that model, but it's good, I think, to have it on the slide there. So those are just my general comments. Next, we have a question to Isaac. And Hussein is asking, what does laboratory analysis of semen involve? And the second question is, besides motility, is there any in vitro measure of semen or cell viability? Over to you, Isaac.
Thank you very much, Hussein, for the question. As he said, laboratory analysis, I will have to check the concentration of the semen, and then of course go ahead to do the mortality assessment, then the hyposmatic test to see the membrane integrity. So I think the second question is answered. Besides motility, I'm also going to look at the sperm membrane integrity, and I also intend to do a DNA analysis to see the changes at those levels. Thank you.
All right. Thanks for the response. Now we move to Enock. And before I read the question, I wanted to ask if any of your supervisors wants to comment. I know Hussein is in the meeting. I'm not sure whether Bernard is still on. Any comments from you, the supervisors?
Maybe not me, maybe Enock. But just to say, maybe Hussein. Enock is part of the project, which is being done by other graduate fellows. And so he takes advantage of the samples which come from those students to do further investigations on Coxiella. So I don't know, maybe Hussein has further comments.
Hussein?
No, I don't have any comments.
All right, thanks. Let me finish the questions on the chat, and then we go to Phyllis. I see you have your hand up. So Eugene is asking to Enock, can Q viva resort to abortion in humans? Enock?
Okay. Thanks, Eugene, for that good question. I think as it stands for now, the effects of Q fever or the impact in humans is that it is only known to cause febrile illnesses, and the symptoms normally resembles the symptoms of malaria.
Okay, I hope that answers the question.
Actually, if I may add, there are reports of spontaneous abortion in pregnant women as well. So maybe it needs to be investigated further, but there are reports.
Okay, thank you for the clarification, Hussein. We have another question to Enock from Lynn. What's the justification for the rooted tree as opposed to other analytical tools? Over to you, Enock.
Okay, thank you, Lynn. So there are, according to my knowledge, there are 3 ways of representing a phylogenetic tree. One way is to use a rooted tree, of which that is what I used in my presentation. And the reason as to why I prefer this is that it will give inference about the most common ancestor. So unlike the other methods, like the use of unrooted tree and the use of bifurcating tree, this one, the rooted tree, will give me an inference about the most common ancestor. Thank you.
Okay, thanks for the response. And Bernard is adding that Coxiella also causes pneumonia in humans. So yeah, that's why I see the need of being careful when handling the samples. So now I move to Phyllis. You have your hand up. Yes.
Yeah. Thanks, Nicholas, and a good presentation to the three presenters. I think I'm just going back to Damaris. So let's say from the literature, I understand your supervisor mentioned that these are uncharted waters, but from your literature review, how do you expect the trends in food security to change in the presence of RVF outbreak? And also considering that mostly RVF occurs after a period of droughts, mostly during the outbreak during like El Nino rainfalls. So it follows as a period when like food security was so dire. So do you expect like it to worsen, to be better? Like what do you anticipate, and what do you anticipate, and what do you want to prove like wrong or right?
Thank you, Phyllis, for your question. Yeah, yes, I can say during the RVF outbreaks, we expect it to negatively affect the food prices, but it's true that these outbreaks occur after an anticipated period of time, especially excess rainfall and periods of flooding. But now to really emphasize on that, the study will also do some retrospective analysis whereby I'll get data on previous outbreaks. That's the 06, 07, another one in 2018, another one in 2020. At the same time, I'll also get from the World Food Program reports on the specific food prices, especially the meat, milk, the foods from animals, and do some statistical modeling to really investigate what happens whenever there's an outbreak, what happens to these food prices, what happens to the availability, what happens to the stability, and all that. Thank you.
And does that answer your question?
Yes, Nicholas. Thanks, Damaris.
Okay, good. I have just two more comments. One to Isaac. Your work is around semen extenders. I just think it's good when you have some of these terms which are like in the middle of your work to explain, not everyone understands what that is. So you could probably mention what that is. And to Enoch, you said you'll use opportunistic sampling. How are you implementing that? You have people kind of reporting and then you go sample, or how is that being implemented?
Okay, thank you, Nicholas, for that question. So samples will only be collected upon the notification of apportion cases by farmers. So farmers will call the field team that there's a case of apportion, and the field team will go and collect the sample.
Thanks. That's clear. To Isaac.
Thank you, Nicholas, for the question. I think I tried to explain some of the terms, maybe what I missed. Don't know, maybe VAP, which is average path velocity, then progressive motility, then the linearity of this the sperm cells or the semen. But I also try to, as much as possible, explain the other terminologies that I have. I don't know if I've explained what I've explained is what you're expecting.
No, that's fine. That's fine. Thanks. I just think generally it's good to make clear, like explain. So don't assume like people know these terms. So any other questions? All right, if there are no more questions, thank you so much to the three presenters, Damaris, Enock, and Isaac. You really did a nice job. Of course, take into consideration the comments raised and keep improving your work. And we wish you the very best as you move to implement your studies. And thank you to the audience for joining and actively participating, and to the capacity team, Malvin and Jennifer, for setting this up, and thanks for the opportunity to moderate. So have a good afternoon. Bye, everyone.